Good afternoon, dear colleagues. My name is Nikita Lomov. I represent the joint team of the Institute of Theoretical and Experimental Biophysics, Pushinsky State Institute of Natural Sciences, Lomonosov Moscow State University, and Federal Research Center Computer Science and Control of Russian Academy of Sciences. I'm glad to present to your attention a talk on registration of regeneration in planarians from photographic images. The object of our research is planarians, flatworms living in freshwater reservoirs. Small size, fast reaction rate to changing conditions, and ease of cultivation made it possible to use a number of planarian species as exemplary objects for biological research. Planarians have outstanding abilities for regeneration. With a transverse section in the head region, both parts of the planarian body survive. The head part grows the tail end, and the tail part grows the head end. Moreover, a planarian can be cut in almost any direction and into a very large number of parts, up to 200, and each of them, under favorable conditions, will restore the missing parts and form a whole organism, provided that there are enough stem cells in the relevant part. Due to this property of planarian, it can be used as a bioindicator of the ecological state of the environment by measuring the speed of the regeneration. The software currently used in Pushino laboratories to work with the images of planarians contains a number of original image processing algorithms, but it works in a semi-automatic mode and requires user intervention. For example, the user is prompted to specify points, the apex of the head and the end of the tail, to measure the length of the worm. Our task is to develop criteria and algorithms for fully automatic estimation of the regeneration rate. The first stage of planarian image processing is object segmentation. For binarization, we use common image analysis techniques: Gaussian filter, morphological opening operation, and threshold binarization. In thresholded image, we choose the largest connective component, but since the vessel walls can get into the frame, an additional test is carried out. Such a component cannot join the frame boundaries. Blastema is the accumulation of homogeneous non-specialized cells of the wound surfaces after organ amputation. During regeneration, new head tissue grows from the blastema. With the course of regeneration, the shape of the blastema becomes more and more pointed, and consequently the area of that part, into which a sufficiently large circle cannot be inscribed, grows. It is proposed to calculate the area of the spot not covered by circles, as well as the distance to the apex of the blastema, depending on the size of the circle, as a characteristic of the regeneration rate. With an increase in the radius of the circle, its center begins to move from the apex to the tail. Since we are dealing with inscribed circles, the concept of a skeleton naturally arises. The skeleton is the set of centers of all circles inscribed in the figure. If I take a simple polygon as a shape model, then the skeleton of such a figure will be a geometric graph with edges formed by continuous lines. This format is very convenient for analysis. You can analytically calculate distances, tangents, curvatures, and so on. The shape of the planarian is very simple. It is well approximated by the union of circles lying on the single branch of the skeleton. To determine this branch, first remove from the skeleton all minor branches, the presence of which has little effect on the shape, and then find the apex of the head and the end of the tail as points farthest from the resulting skeleton. To ensure the scale invariance, the proposed criteria are normalized by the radius of reference circle, the circle that is located In the region of planarian's neck, we see that for a single planarian, both proposed criteria increase in the course of regeneration. However, in order to obtain informative assessments, it is more correct to consider not individuals separately, but groups of individuals. In addition, individual planarians are difficult to keep track on over time, as their natural habitat is collective. Single planarians regenerate worse and also tend to split into a pair of individuals themselves, discarding their tail. Therefore, for this, the individual estimates obtained are averaged over the group. In the experiments, 3 groups of 20 planarians were compared. An experimental group consisting of planarians placed in water, and 2 test groups placed in solutions of a peptide with a concentration of 10 picomolar and 10 nanomolar. They were photoshooted for several days. Results for different groups of the same day as well as results for the same group on different days were compared using a one-tailed t-test. Thus, the very fact of regeneration of planarians over time is beyond doubt, and the proposed criteria record the fact of regeneration for all 3 groups. Also, the experiments reflect a statistically significant difference between the control and each of the test groups, at least on the second day, taking into account the correction for multiple hypothesis testing. The main direction for further research is an identification of a single individual. It is proposed that the type of hat, the shape and its color differs quite strongly for different individuals. However, this approach is only suitable for full planarians that haven't undergone decapitation. For headless planarians, it's suggested to analyze light spots, areas without pigment located throughout the body. It is argued that the shape and location of such spots is stable for at least several days and is individual for different planarians. As a result, a fully automated system for assessing the degree of regeneration of planarians was developed based on the analysis of the skeleton of a binary image. The experiments carried out confirmed the possibility of stimulating regeneration with the help of a peptide. Thank you for your attention.